Solutions, Part 2, Osmosis, Plasmolysis, and Imbibition. This is the movement of water from a region of higher water potential to a region of lower water potential through a selectively permeable membrane. The driving force of osmosis is the free energy difference between the two regions of water, which is equal to water potential gradient. Osmosis helps in the absorption of water by plants. Movement of water from one cell to another is due to osmosis. Osmosis brings about the opening and closing of stomata. Osmosis can be demonstrated by experiments like thistle funnel experiment and potato osmoscope. Potato osmoscope. Peel a raw potato and make it into two halves. Scoop a piece out of one half and pour sucrose solution into it. Place the scooped potato half into a glass plate with water. After some time, we can see the rise in the level of sucrose solution inside the potato, confirming the fact that there is the movement of pure water through the potato, which is acting as a selectively permeable membrane. Thistle funnel experiment. A thistle funnel with a broad opening below and a narrow opening at the top is taken. An egg membrane, which acts as the selectively permeable membrane, is fixed to the broad mouth of the funnel. The funnel is filled with sucrose solution up to a level in the tube. The inverted funnel is immersed into the beaker with water. After some time, the level of the solution in the tube of the funnel rises due to the movement of the water through a selectively permeable membrane. We can say that energy flows from higher level to lower level, thereby moving the pure water from the beaker into the thistle funnel. As the two systems reach an equilibrium point, a pressure known as hydrostatic pressure is developed in the thistle funnel. Plasmolysis. The shrinking of protoplast of the cell due to loss of water when placed in a hypertonic solution, that is, high solute content than the cell, is called plasmolysis. This condition, where cell exhibits plasmolysis in the corners of the cell wall, is called as incipient plasmolysis. The loss of water from the cell due to higher water potential inside than that of the outside is called osmosis. In a plasmolyzed cell, the turgor and wall pressure remain zero. Osmotic pressure contributes to the water potential. A plasmolyzed cell, when kept in a hypertonic solution, regains water by osmosis. and the process is called deplasmolysis. If a solution is kept in an isotonic solution, the solute concentration of the solution is equal to that of the cell. There is no net diffusion of water taking place. The salting of pickles, addition of sugar to jams and jellies, plasmolysed bacterial and fungal spores which spoil the food. Imbibition. The absorption of water by hydrophilic colloids is called imbibitions. Example: dry seeds exhibit imbibitions before germination. Proteins have a very high imbibing capacity when compared to starch. That is, proteinaceous pea seeds weigh more than starchy wheat seeds due to imbibitions.